Welcome everybody to today's edition of the Extracellular Vesicle Club. This is a weekly event of the International Society for Extracellular Vesicles, and it's my pleasure this week to introduce our two guests, Eva Zuba-Surma and Ela Karnas, who are joining us from Poland. And in fact, they have just recently started a new national society, the Polish Society for EVs, which we just heard a little bit about before we started. And Eva, welcome. I'd like you to just perhaps tell our guests how they can contact PSEV if they would like to get involved.
Yeah, so basically we have an email address and on the contact information, which we'll be happy to share after this meeting. We can spread out this information via ISEV website or directly. If somebody is interested, we can share the information. The best would be to contact directly me via email or Ella, who is also involved in the board of the society, and also will be happy to share the whole information, how to join and how to be a part of society.
Wonderful. Well, thank you for that. And do you mind, may I put the email addresses in the chat box today?
Sure.
Wonderful.
Sure.
Okay.
I would be happy to provide, yes.
Yeah, I'll do that. All right. Okay, well then let's go ahead and get started. We want to get to your very exciting work on iPSC derived EVs and what you're able to do with them. So please go ahead and start your screen sharing.
Thank you very much. I think the screen should be already visible. Please, if you can confirm.
It looks good.
Everything is fine. Great. So thank you very much, Kenneth, for the very kind introduction, also for introducing us and our society. And I would like to welcome everybody. I'm very happy that so many participants join us today. Thank you for that. And first of all, thank you, Kenneth, for inviting us for this seminar and giving us opportunity to share the recent project, the results of our research project, which is focused on extracellular vesicles derived from iPS cells, from induced pluripotent stem cells, which I believe are quite well-known stem cell population for community, which we recently envisioned as a potential novel tool to boost hematopoietic properties of human cord blood cells. And the paper which we are going to present, it's a major work of Ella, who was also introduced by you. She's a brilliant postdoc in my lab who is working with me for last several years and actually gaining experience in the stem cell field and EV field. Along with me right now, she's practically my right hand in a work with extracellular vesicle characterization, isolation, and also looking at the molecular properties and functional properties of EVs in various in vitro and in vivo aspects. So I'm really happy that I can present today this project, which was really her PhD project conducted for a few years. Like last week, we had an Clotilde Terry group also presenting extended paper done during the few years of work, and the work that we are presenting today is a very similarly done for multiple months, very precise work. And we believe that this data, and I hope I will be able to convince all of you in the audience for that at the end of this presentation, we believe that may have potential implications for future use of cord blood units in the clinical practice, because as we know, there are several cord blood units which have been stored in multiple cord blood banks worldwide, and we know that they are currently quite underutilized because of some limitations, which I will talk about in a minute. And we hope that the data that we just published in the recent paper may open some new perspective for the better utilization of this very precious material in the future. So before I will move to the, there is some Oh, okay, sorry. Before I will move to the paper itself, let me introduce briefly my group and the work that we are doing in Krakow, the Jagiellonian University, and also the data very briefly, which brought us into the point that we focused on the hematopoiesis and the use of EVs from induced pluripotent stem cells for enhancing hematopoiesis. So this slide, you can see my group at the JU, and all together for last few years we are focusing on the studying extracellular vesicles from stem cells, mostly from the mesenchymal stem stroma cells of various origin, and the induced pluripotent stem cells that I just mentioned, because these are the major actors of our recent paper, so as a source of extracellular vesicles. And as we know, they resemble embryonic stem cells. And right now in the stem cell field, iPS cells are envisioned as a potential optimal source of cells for autotransplantations. However, we should remember about the, let's say, the second side of the coin of these iPS cells, that they also harbor the teratogenic potential as a pluripotent cells. So in our group, we wanted to verify whether iPS cells can be also envisioned as a source of extracellular vesicles that represent in our hands, in the regenerative medicine, in stem cell biology, the arm of the paracrine activity of stem cells. So we wanted to know whether maybe iPSs can be better utilized, safer utilized for future repair. And in our group, we really indeed evaluated the composition, molecular composition, and also properties of extracellular vesicles from iPS cells in several in vitro and also in vivo models. And we published already in 2015 the very first paper. Actually, that was the second paper worldwide, so very early, showing the scientific audience the new concept that iPSs may be considered as a source of extracellular vesicles, which we may use in a safer manner to stimulate pro-regenerative effects in injured tissues. And in this paper published in Stem Cell Journal, that time we focused on the cardiovascular repair because that's our primary field. But in this paper, we very well characterized molecular cargo of human iPS-derived extracellular vesicles, showing that they carry several microRNAs that, of course, regulate not only pluripotency, because they are pluripotent cells, but also they may regulate the cell differentiation, cell behavior, also potentially hematopoietic properties of cells. And also we found several proteins that can be involved in regulation of tissue development, so also potentially hematopoietic tissue development and regulating several signaling pathways. And of course, cord blood, we are very much interested in this material because it has been for a long time envisioned as a potential alternative material, clinical material for the bone marrow, for mobilized peripheral blood, as an enriched source of CD34 positive hematopoietic stem in progenitor cells that can be used potentially to reconstitute also effectively hematopoietic system. And indeed, as I mentioned at the beginning, we have right now several cord blood units stored worldwide that are waiting to be really well utilized for the clinical practice. Why? Because there are several benefits to use potentially cord blood units. Cord blood cells collection is non-invasive. The cells are less frequently provide graft versus host disease, and also lower HLA match is required before donor and recipient when we envision this cord blood derived cells for transplantation. However, also as I mentioned, there are unfortunately some limitations which do not allow us that well use the cord blood for the clinical practice, and one of them are the limited number of hematopoietic progenitors in a single cord blood unit, which is absolutely the limit if we want to transplant such a unit for some adult patients. And of course, the delayed engraftment in the bone marrow in adult patients is also one of the obstacles. That's why we start to think whether, knowing all the properties of extracellular vesicles that we study, we can propose maybe some novel strategies how to overcome these obstacles. So limited number of the hematopoietic progenitors, and also delayed, so a little bit limited hematopoietic properties of this cells. And of course, we know that stem cells may produce extracellular vesicles, that they can carry the bioactive components into the target cells. So they are important elements of cell-to-cell communication. I don't need to explain that in this audience. We all know there is a growing number of the papers showing that the stem cells are valuable sources of EVs, impacting also on the functional properties of the target cells. And from our work, we know that iPS cells, these induced pluripotent stem cells, they can produce extracellular vesicles that can be safely utilized not only in vitro to change the phenotype of the cell function of the target cells, but also in vivo. So they can be envisioned as an alternative for the using of cells for transplantation. So that's why we ask the question whether we can also impact on the functional aspects of the cord blood hematopoietic progenitor stem cells by stimulating them or incubating them with the iPS-derived extracellular vesicles and impacting on the function that are important from the perspective on effective engraftment and bone marrow reconstitution. And this is the story which brought us into the most important aspect of today's seminar, so presenting the data recently published. So the data that is actually answering this question. We are very happy that, yes, indeed, iPS-derived extracellular vesicles may enhance this biological properties of cord blood cells. And we believe that when we optimize, we understand this phenomenon better, we optimize the strategy, maybe we can offer something for the better practical use of those cells. And as I mentioned, it was published recently in Leukemia. It is open access paper. We decided for that. So everybody are very welcome to download the paper, to read it, and to look into the details of the paper. I will just show few selected results from the paper to just draw you the concept of the study and the major results. As I mentioned, I would like to once again acknowledge Ella, who did really the great job and a lot of work she put into that because on one hand, she worked with extracellular vesicles from iPSs, which is quite difficult field. And on the other hand, she worked on the highly purified populations of cord blood cells, which also required a lot of work and a lot of material. So coming into the paper itself, we have two major actors in this story. So one is our extracellular vesicles that we isolated from the serum-free conditioned media from iPS cells. So that was completely serum-free system, xeno-free system as well, to have highly purified enriched iPS-derived extracellular vesicles that we, in this study, isolated via sequential centrifugation, including ultracentrifugation step. And the second actor are hematopoietic progenitor and stem cells derived from the cord blood, which were isolated via three-step isolation strategy, including red blood cell lysis, followed by the magnetic sorting based on the CD34 expression, which in the hematopoietic field is one of the major markers related to the stem cells, hematopoietic stem cells. And then the third step included the FACS sorting based on the already quite extended phenotype of the cells. So we focused on the lineage negative. Lineage means the cells that did not express the major hematopoietic system markers. They were not differentiated into major lineages of hematopoietic system. CD45 DIM means CD45 is a pan-leukocytic marker, which would be present on the majority of hematopoietic lineages. The cells were dim, so they were very early progenitors expressing CD34 antigen. So for you, so that you know exactly which target cells we used in this study. And we always use such a strategy when we focus on the highly purified populations, relying not only on the max, but always on the FACS sorting as well, to make sure that our population is highly pure, as in this case, when we had close to 99% of the purity of our cells. And of course, starting our characterization, before characterization, I'm sorry, I will just mention that we use the 2 treatment protocols of this cord blood hematopoietic cells in the long and short-term incubation protocol. So long-term was mostly focused on the examining of impact of extracellular vesicles on the expansion of the cells, whether we can use them as a valuable element of an expansion media. So we use the EV presence during the whole expansion for 14 days of the cells, and we perform functional tests, and short-term incubation to just see whether very short, 2, 6, up to 48 hours incubation of freshly isolated cord blood cells would have an impact on the gene expression, on the activation of some signaling pathways, and on some other functional aspects which we studied. And coming to the results, of course, in the first step, I will just move faster through that. We, of course, follow the recommendation of ISEV and ISEV guidelines to characterize the EVs that we isolated. So we visualized them by atomic force microscopy, also performed the size distribution and concentration analysis by NTA. We have a nanoscience N300 in our lab. And also Ella has performed a very extensive phenotypic analysis using the high resolution flow cytometry, the apogee system, and also image stream system, focusing on several different markers present on these EVs, considering both parental surface markers as well as, of course, exosomal markers, including tetraspanins. And just selected results shows that majority of these vesicles, about 75% of them, they contain RNA, as shown by the positive staining with RNA select. We consider them as an integrative vesicles, and majority of them express also parental markers and also tetraspanins, as shown here. And with this vesicle, our samples which fulfill the criteria, the definition by ISEV, we moved to the further studies. So we incubated the hematopoietic cells from the cord blood, and we confirmed that yes, indeed, they are interacting with the extracellular vesicles, which were in this case endogenously stained with COP-GFP. We confirmed they are internalized. So then we focused on the functional assays. So we wanted to first find whether there is any impact on the proliferation. And I hope you are not surprised, because as I tried to introduce you into this concept of enhancing properties of cord blood cells, first would be the beauty, if that would work, would be to confirm that there is an impact on the proliferation. Then we would have a beautiful protocol that we can use the iPSEVs to enhance proliferation, to expand the hematopoietic progenitors from the cord blood in vitro, and just simply have a greater number of these hematopoietic precursors in our hands so that we can use them in humans. Unfortunately, in our studies, we did not see the impact on the proliferation. So yes, indeed, there was an ex vivo expansion, then the fold expansion changed significantly. However, there was no difference between the control cells, means the cells that were expanded in the expansion, dedicated expansion medium, shown here as an open circle, versus the cells that were additionally treated during the expansion with the extracellular vesicles. However, when we briefly exposed the hematopoietic progenitors on the extracellular vesicles, we showed the increase in the metabolic activity. That was very interesting observation that ATP concentration significantly increased after 24 hours after such treatment. So that made us thinking that those cells may actually start maybe differentiation process during this expansion. And that was actually quite going along with the change in the expression of CD34 antigen in this culture. As you may appreciate, the cells proliferate, they expand, but in the same time, the percentage of the cells expressing CD34 antigen, so representing the most primitive, the early developmentally hematopoietic progenitor, decreased. So that could indicate the spontaneous differentiation of those cells, although we used very highly dedicated, very well-defined, commercially available medium that is dedicated for an expansion of early progenitors. But you may appreciate that expansion goes well up to 8 days, then the cells drop in the content of CD34 antigen, means the cell starts differentiate. And that was confirmed actually by much more deeper analysis performed by Ella, by flow cytometry. So she looked, along with this decrease in CD34 expression, she looked on different populations of potential hematopoietic cells giving rise from these progenitors. So looking at the CD3, CD14, 19, 66B, CD20, 35A, so all the markers of different lineages of hematopoietic system. And indeed, she found that there is an increase in the populations of those cells during this expansion. So that means the cells spontaneously start to differentiate. And indeed, interestingly, those population, this population that was treated or expanded in the presence of EVs, they spontaneously differentiated in the greater manner when compared to the control cells. So that brought us to the point to check whether there is any enhancement in clonogenic potential, which is very closely related to the differentiation capacity of hematopoietic cells. And we checked whether with this decrease with the primitivity CD34 antigen, there is any change in the clonogenic potential. And we found that yes, indeed, although there is a little bit drop of the cells that would form the different hematopoietic colonies, which you can see on this upper panel, but still we can see this very important increase in this clonogenic potential when we treat these progenitors with extracellular vesicles. And this phenomenon was also present when we treated just briefly, shortly, 2, 6, or 24 hours, the freshly isolated progenitor. Still, when we checked the clonogenic potential of the cells, then we found that it was significantly increased when the cells were treated with extracellular vesicles. As you can appreciate, these are beautiful pictures taken by Ella. We typically focus on different types of colonies in hematopoiesis because all of them represent a little bit different lineages of differentiation. So here we have a total number of these colonies, but we also looked separately, counted these colonies, and this phenomenon of the increase in a colony forming units after the treatment with iPSEVs was actually present in all types of the colonies that are here. I provided you exactly the description, what each abbreviation means in hematology. So pretty much granulocyte macrophages colonies, erythroid colonies, and the mixed colonies were increased after the treatment of this progenitor with iPSEVs. So that part of the study provided us an evidence that the treatment with iPSEVs, of course, glue is a molecular cargo, which is inside of these vesicles, may increase the clonogenic potential, which is very important for the reconstitution of the bone marrow system following the transplantation. However, we have to be aware that clonogenic potential of hematopoietic cells is not the only functional feature, which is important for the reconstitution of the bone marrow system. And another very important function is the hemotactic activity of the cells, because we have to remember that when we transplant the cells, they need to find the way. So they need to home into the bone marrow niche. When they will be anchored, they start proliferation, differentiation, and finally they will constitute the system. So we looked at the hematopoietic hemotactic activity using the in vitro model, using the Boyden chamber test, when we put our cells, the progenitors from cord blood, into the upper chamber, and the hemotactic agents, which was SDF1, stromal-derived factor 1, into the lower chamber. I have to admit that SDF1 is a major chemokine produced in the bone marrow system by the mesenchymal stromal cells that would attract the hematopoietic progenitors into the bone marrow niche. And in this system, these two parts of the chambers are separated by the membrane with the 5-micron pores. So the cells cannot freely go into the lower chamber, but they have to be attracted by the hemotactic agent. And then we count how many cells are responsive, and they are located eventually at the end in the lower chamber. And making long story short, found significantly increased number of responsive chemotactic cells in the lower chamber after 2 and 6 hours of treatment with extracellular vesicles when compared to control. So the cells that were not treated with extracellular vesicles. And we found surprisingly that it was not associated with the level of an expression of CXCR4, which is a receptor for SDF1, but in our opinion, it was rather related to the robustness in the activation of the signaling pathway related to this receptor and SDF1, which Ella have shown by a very nice assay, which she actually optimized completely for this study with measuring the calcium flux. Calcium is a secondary messenger, very important secondary messenger that is important in the signaling pathway. And when the cells were treated with extracellular vesicles for 6 hours, she showed the significantly higher influx flux in such cells. Another very important feature, which is also involved in the effective reconstitution of the bone marrow system, hematopoietic system by hematopoietic progenitors, it's an adhesion to the niche. And in our study, we used the model elements of hematopoietic niche, including the fibronectin representing the extracellular matrix protein, and also two cellular components, artificial components, let's say hematopoietic niche, including mesenchymal stromal cells and endothelial cells such as HUVECs. And making long story short, we incubated our calcine-stained hematopoietic progenitors, exposed or not exposed to the iPSEVs with these different components of stem cell niche. And we found again that also adhesive properties of the cells are increased following the treatment with iPSEVs in all the situations of the fibronectin, MSCs, and into HUVECs. And Ella was looking for explanation of this phenomenon, looking at different adhesion molecules present on the cells, whether this iPSEV stimulation will change the level of expression of several antigens. And she found, for example, that one of the antigen FLA1, which is a ligand for ICAM1, so the adhesion molecules, which is very well known in intercellular interaction, was significantly increased on the cells that were treated for 2 or 6 hours with extracellular vesicles. Of course, it's just a selected results. This phenomenon, which needs to be a little bit more deeper explained and investigated, which we are going to do, because we envision that this phenomenon would be probably related to the change in the expression of some more molecules present on the progenitors following the treatment with EVs. And another very important aspect, which cells will need to, let's say, undergo when it is anchored in the niche, is the survival. So that's why we also looked on the level of apoptosis, how the hematopoietic progenitor will sustain the cytotoxic environment when they are treated, in this case with staurosporine, in the presence of extracellular vesicles. And indeed, we found that when the cells are pre-treated, primed with the EVs, and then exposed into cytotoxic environment, which can be present, for example, into yellow-obliged niche, they are greater in their survival. We found a much greater percentage of alive cells, which was accompanied with the lower early apoptosis level in this population. So all this part together showed that not only clonogenic potential, but also other functions, including hemotactic activity, including adhesion properties, and also survival of the cells is increased following the treatment with extracellular vesicles. And eventually, Ella asked the question, what can be the mechanism beyond that phenomenon? What can be responsible for that? And to answer, at least generally, for this question, we checked the change in the expression of several genes related to the hematopoiesis. This was an array that was targeting the expression of several genes related to the hematopoiesis in the cells that were treated with extracellular vesicles, actually were expanded in the presence of extracellular vesicles when compared to the ones that were expanded in the classical medium and the ones that were just freshly isolated. And I'm sure you can already appreciate that several these genes related to hematopoiesis were upregulated, especially 4 days after expansion in the presence of extracellular vesicles. And all of these genes are described and discussed also in our paper with details. So I will just focus on few of them. We just selected from all of this heat map few important genes that are involved in hematopoiesis, including transcription factors, RUNX, as well as Notch1, Notch2 PAX. So all of this were upregulated after the treatment with the EVs, and also further bioinformatic analysis confirmed the interactions, the network interactions between those genes that we found as upregulated following the treatment with EVs. And also in the next step, we checked whether short stimulation, quick stimulation with extracellular vesicles, just 2-hour stimulation, will have any impact on the signaling pathways in the cells. And yes, indeed, we found that several signaling pathways regulating hematopoiesis and function of the cells, proliferation, survival, regulation of the immune response, which is related to the also maturation of cells, they are changed after the treatment with EVs, which was shown by the phosphorylation of several kinases that may be involved or were described to be involved in hematopoiesis. Again, it is described well with details in the paper, but I just show here stats or other MAP kinases that are involved in hematopoiesis. They were greatly upregulated after the treatment with EVs. So all of this together brought us into the point of the question whether this phenomenon would have any impact on the homing and engraftment of the progenitors in vivo. So we performed a short in vivo or just a small in vivo study where we used expanded cord blood cells primed shortly with the extracellular vesicles from iPS cells. And then as a recipient, we use the non-skid mice that may accommodate the human cells, and we divided them into 3 groups. One group obtained the hematopoietic progenitors not untreated. The second treated with iPS EVs, and of course, as a control, we used the vehicle. And we evaluated homing and engraftment after 48 hours and 8 weeks following transplantation, respectively, looking at the content of human cells in different tissues, including peripheral blood, bone marrow, and the spleen. And again, just making long story short, looking at the homing and 48 hours following the transplantation, we found significantly greater number of human hematopoietic cells in the murine bone marrow niche. It was a slight increase in the spleen, but it was not significant, and also it was associated with a slight decrease in the peripheral blood. Of the cells in the peripheral blood, which is quite logical, because this is what we would expect. If the cells would home into the bone marrow, we would expect to have a little bit lower concentration of the cells in the peripheral blood. But more importantly, after 8 weeks following transplantation, we found significantly increased number of human cells, transplanted cells in the bone marrow, in the spleen and peripheral blood, in case of the group that obtained the hematopoietic cells pre-treated, primed with the iPSEVs. So that very initial, very preliminary experiment, but very important for us, because confirmed that yes, this phenomenon can be also visible in vivo when we have enhanced engraftment of hematopoietic cells, cord blood hematopoietic cells, after the priming with iPSEVs. So to sum up and conclude, in our paper, although we did not show the effect of iPSEVs on the proliferation rate of cord blood hematopoietic cells, but we show several other aspects that were increased following such treatment, including impact on the metabolic activity, survival, and more importantly, on the clonogenic potential, hematopoietic activity, and adhesive properties, which may have a direct impact on the efficacy of the hematopoietic system reconstitution following the transplantation, which was also confirmed by this in vivo study showing the increase in the homing and engraftment of the cord blood progenitors treated with extracellular vesicles, and the mechanisms which we are still evaluating, and we will be following this project to deeper look into the mechanisms responsible for these phenomenons, we believe are related to the expression, the activation of the gene expression related to hematopoiesis, and also to the activation of some important signaling pathways related to hematopoiesis, which I have shown you. So I believe that we will continue. I hope also Ella will continue that project with the new study, where we can optimize this approach to propose more optimized protocol for preconditioning of hematopoietic cells from cord blood with the iPSEVs, to bring these cells again a little bit closer to the practical applications to benefit patients. And at the end, I would like to acknowledge once again my group, especially Ella. As I mentioned, this is really her project, her work. Other people working in my group and also in the department, as well as other collaborators who were involved in this particular project focused on hematopoiesis. And thank you once again for your attention, for your attendance, and I will be happy to answer your questions. Thank you.
Thank you so much for this opportunity, and it was a very nice work. Even I have presented this paper in my journal club. Everyone is appreciating. So my question is that what is the passage of iPSCs, for example, which is the passage number? Do you think it's optimal to collect the extracellular vesicles?
So I'm not sure if passage would be that important for EV collection, because in terms of iPSs, we work with pluripotent cells. So they are not losing that much their pluripotent properties that would impact on the cargo of extracellular vesicles between passages. So we are using iPSs on various passages, And when you compare the content of the EVs, when you use the same cell line in the same culture conditions, is quite similar. That would be very important question if you would have other cells, other stem cells like mesenchymal stem cells, for example, because that would matter very much depending which passage you use, because that cells will just undergo aging with time, with passages going eventually into senescence. So earlier, the better, of course. But in terms of iPSs, the passage is not that crucial for the quality of your EVs. Very important would be the confluence and the comfort, what I call comfort of the cells. You always need to pay attention to make sure that your cells would be comfortable, would be healthy. So we always check the apoptosis level. We evaluate the overall conditions of our cells, because that would significantly impact on your EVs. If the cells would be unhappy in the confluence after the passage, after sealing, then of course you will not get a good yield and good molecular quality of your EVs. We work with, as I mentioned, with serum-free medium. So that costs us also a lot of effort to make our iPSs to grow completely in a serum feeder layer free conditions. So that is also something which makes your EVs more standardized. So if you are interested, I would recommend also to move into the serum-free medium, which will make your quality of your EVs greater and more comparable between the preps. Yeah. So thank you. I think you put your question on the chat, right?
Yes.
Because I could not hear you very well, but I can see your question in the chat. So upregulating of adhesion molecules is exciting. Yeah. So basically, the expansion of our cells we perform in a highly standardized recommended medium that is on the market, which is a stem spam SFM2 medium from the stem cell technologies. So of course the composition of this medium is unknown to us, but this is a very much recommended medium for the expansion of early hematopoietic progenitors. So I believe, of course, there is some growth factors. It would not be possible for the cells to be expanded without any cytokines. So I'm sure there are cytokines there, but we don't know what cytokines, what concentration. We know the concentration of growth factors in methylocellulose, for example, when we are culturing the cells, but it's not expansion, it's more like differentiation. So this clonogenic assay, this is much more unblinded for us because we know exactly concentration and all of these factors that you mentioned in your chat post, that are present in the methylocellulose when we do the clonogenic assay. But for the expansion, we have a commercially used medium which we don't know the exact composition, but I'm sure there are cytokines there.
Thank you so much.
You're welcome.
Our next two questions, I am going to summarize right now, but they come from Raji AR and Louis Vu. So please feel free to unmute yourselves and expand on what we're discussing here. But let me just summarize. The first question is, will there be any secretory proteins in the isolated EV preparation? And what would be the best way to remove those secretory proteins? And the second question, I think, which comes along in the same vein, is about a so-called process control. So have you ever tried to just take the medium that has all of your factors in it and put that through the EV separation process and see if anything happens with just those elements that might survive the separation in the medium itself?
Thank you. Thank you. Yeah, so that is always very important question about the quality control. As I mentioned, we use the serum-free medium. So at least we believe we do not have components from the serum, which is a major impurity when we consider isolation of EVs from the conditioned media. Of course, there may be still some proteins, and we know because we did some proteomic analysis that there will be some significant presence of BSA, for example, because in serum-free media you have to have some income. So instead of FBS, we have BSA, for example. But BSA, it's a ballast for us, but we believe it would not be the functional ballast. The problem would be the cytokines, which would be, and I believe Raji is more considering this, that maybe we can isolate some cytokines along with EVs. We believe that will not be that many such impurities because we purify those EVs, we wash them a few times, but of course, we cannot exclude that there is no some impurities considering proteins there. In this study, we used ultracentrifugation, so of course we cannot exclude that something, although we purify, we wash the EV specimen, still is isolated. Of course, we could use some additional column purification, which we are trying to use in other studies, but in the study we did not use it. We did not use the control with the full medium. Simply, we had the discussion many times about that in our group, because we believe that the cells will simply metabolize this medium. So that would never be a perfect control for us. If you would use the full expansion medium, although it is a serum-free, but it has a bunch of stuff inside which needs to sustain, provides the viability for the cell expansion, for iPSs especially, which are very, very sensitive cells, and this is serum-free medium, then you would have probably great impact on the other cells with such a medium. But we have to also understand that iPSs, they are very fast metabolizing cells, and they will metabolize this medium. So we have not used that. Although you are absolutely right, we did not use this control, but we could. Maybe that would be a good idea also to consider that in the future, to use, for example, some supernatant or EV-free supernatant after the processing. That would be maybe better control. So this, like the rest of the conditioned media, already metabolized by the cell, that would be maybe closer to the situation if we have something, I don't know, co-purified with the EVs from this conditioned media. But in this case, we did not use any such a control, which you mentioned.
Good. All right. Thank you. Let's go to Susan Goals next. Susan, did you want to tell us about VLA4 and why you're asking about that?
Yeah. VLA4 obviously is an adhesion molecule that occurs on most lymphocytes, but it also occurs in leukocytes, well, not neutrophils. But when you treat MS patients with a VLA4 antagonist, an antibody against VLA4, one of the things that you see in the blood is a profound 7-fold increase in the CD34 positive cells. So it seems likely that the tethering of those CD34 positive cells in the bone marrow may be to some extent mediated by VLA4. So I was curious about, you tested LFA1 in terms of an adhesion molecule. Did you test also VLA4?
Yeah, we did. We actually tested for CD49 D and A and E. And we have not seen the increase in the expression of these adhesion molecules. So this two adhesion molecules are in the portfolio of adhesion molecules which Ella tested. Because yeah, we believe that maybe that may be also a phenomenon that you mentioned, but no, the only adhesion molecule that we found to be increased following the treatment with the EVs were this the one FLA1, which I showed in the presentation. So if you look into the paper, yes, we mentioned that these two molecules were also tested, were analyzed, but we did not see increase in the cells.
Okay, great, thank you.
Thank you.
Let's go next to Hernando Del Portillo. Hernando, welcome.
Thank you so much, Kenneth. Thank you, Anna, for the talk, and Elizabeth for your work. I have a couple of questions. We have a lot of confounding factors due to the specificity of what EVs does to different cells. So I'm wondering if you have used EVs from other type of cells like MSCs in order to show that what you are seeing is only on a specific of iPS cells. That's the first question. That is the first one.
Yeah, so that's of course a great question. That would be great to do that. We have not done in this study. We work on the MSC EVs, of course. So we have a study when we compare side by side MSC EVs, for example, because I can see in your question on the chat that you actually mentioned MSC specifically as a control. So that's why I'm referring to MSCs. And not in this study, but in other studies, we can see there is a significant difference between activity of these two types of extracellular vesicles, and we characterize them as well. As I show you at the beginning, these are our two major fractions we work with, and we can see great molecular differences between them. And also, if we use them particularly in other study, to stimulate angiogenesis in the limb ischemia in vivo model, we can see completely different activity of MSC EVs and iPS EVs. So I would speculate, because we did not did in this study such an extensive comparison. That would be beautiful, I agree, but that would be another 5 years of Ella's work probably, and isolating another and
Thanks.
using another bucket of cord blood to do this work.
I know the difficulties. Thank you for the answer. The second one is a much more a question that we have, because we are engrafting NSG mice with CD34 cells obtained from cord blood, and we are having a lot of difficulties in getting the engraftment and going to erythroid differentiation. So I'm wondering if you have checked the expression of CD71 or other hematopoietic marker in your homing experiments in the bone marrow with mice.
Yeah, so I think we have not checked exactly the lineages, but Ella was performing the clonogenic assay. So I'm not sure, Ella, if you have this data in front of you, and you can comment on that, because we have not done the evaluation of the exact phenotype, so that the lineages by flow cytometry, what types of hematopoietic lineages we have, but clonogenic assay was done for sure after this engraftment. So Ella, please take a look on the data. I did not include this into the presentation, whether there was an erythroid colonies increased after this engraftment. If we have, that would be probably the answer for Hernando. And if not,
actually, for sure, as far as I remember, because we, as Professor said, we didn't show these results, but we also took the bone marrow from these engrafted animals and performed this clonogenicity potential assay. And we could see still all of these different colonies, including erythroid ones. We haven't done the phenotypic analysis. We focused mainly, as Professor said, it was like a preliminary in vivo study. So we just wanted to see if we have generally hematopoietic human or from origin hematopoietic cells in those mice still present, so originating from our transplanted cells. but we haven't done this phenotyping experiments on these transplanted animals. But as far as I remember, for sure, there were also erythroid colonies. And generally, after this in vivo as well as in vitro studies, we could see increase in the number of all the types of the colonies after iPSEV treatment. It wasn't only effect on, for example, granulocyte colonies, but on all of them. So it was also in terms of this erythroid colonies that you mentioned.
Okay, thank you very much. This is quite important in the translational aspects of erythropoiesis, no? Because this is a major drawback in human erythropoiesis ex vivo and in vivo. So if these iPSEVs can have this effect, this will have a profound, translational aspect of. Thank you so much. Okay.
So thank you also, Hernando, for giving this feedback. That's also very important for us. So we will take a look on this data, and if you are interested, you can be in touch with us. We'll be happy to take a look into raw data and see, especially these erythroid colonies. What I was actually surprised also, there was quite a large number, high number of these mixed colonies with megakaryocytes. And I'm sure you are familiar with hematopoiesis, that also the thrombocytosis, I mean the megakaryopoiesis, it's sometimes very problematic as well. And in our hands, after this iPSEV treatment, we saw quite high number also these mixed colonies that include megakaryocyte development. So we can be in touch and take a look into raw data, because the EVs, they have a heterogeneous composition. That's the point. So these are not like a single molecule, single agents, as all we know. This is a heterogeneous composition of several molecular agents. So now it is a challenge for us to find which all of these molecular pathways will be the most important. And maybe, thank you for pointing that, maybe that will be important to take a look which ones will be especially stimulating erythropoiesis and especially stimulating megakaryopoiesis. So that provides an advantage of using molecularly heterogeneous iPSEVs rather than a single purified agent, right? So thank you for that.
Great. Well, we're coming to the end of our hour, but I'd like to get our last question from the chat box in here. And this is from Meyer Kalma. Meyer, do you want to go ahead and ask this?
Yes, hello.
Hi.
Yeah. My question is, it's about the CD34 species. So if you purify CD34 species containing subset of cell populations such as long-term HSC, short-term HSC, the progenitor HPCs, and other cell lines, will these have no effect on the level of surface marker expression in clonogenicity assays, particularly in the short-term assay?
Yeah, thank you for these questions. No, in this case, we just work on the whole population. We did not focus on the more highly purified subpopulations that would require separation of all this major fraction, which I defined as lineage negative, CD45 dim, CD34 positive cells into more subsets. So no, in this case, we have a heterogeneous population enriched in the CD34 positive cells, which, as you noticed, would include the progenitors that will be a little bit more advanced, and also this very, very primitive long-term repopulating hematopoietic stem cells. But so we did not work on the separate fractions, just on this population, which I mentioned as a general population enriched in CD34 positive cells. And I'm sure if we would go into such fine-tuned study, which would be probably very interesting, that would require a novel project, probably we will see some differences. This is different biological properties between this subpopulation of long, short term repopulating hematopoietic stem cells. So long term will be more quiescent. They will require different stimuli, I believe, to start proliferation, to start the whole hematopoiesis. So I'm sure the impact of the iPSEV will be completely different on these different fractions. So coming to the principle of your question, we don't know which subsets of this population is really responsive, and we are just looking globally because this is also bringing us a little bit closer to the clinical practice, because as you know, in the clinical practice mostly we isolate hematopoietic progenitors just based on the CD34 antigen, if using Clinimax, for example. So we did not want to go into such a fine-tuned population, but more work with the general population. And we use this FACS sorting, additional purification, just for the scientific purposes, to make sure that we have really highly purified population. Because unfortunately, although MAX is recommended as a separation method, in our hand, from our practice, we see that the purity of the CD34 positive cells is not very high. And I'm sure we would have a lot of biased results, which will not be really reliable if we would have unpure population just after MAX. That's why we use the FACS with these additional markers, lineage markers, negativity, CD45 positivity, but DIM positivity, to make sure that our cells are 99% pure, not just MAX. But the principle is to bring this experiment as close to the clinical situation as possible. And in the clinic, we cannot use FACS. We cannot just separate very highly purified population. We just relied on the CD34 antigen expression.
Well, I think that the clinical application is certainly what we're all aiming for. And on that note, I would like to ask Ella a question, because you've worked with that bucket of blood in the lab, and you've worked with these massive towers of cell culture flasks, and ISEV is having a meeting at the end of the month, a workshop that's on massive production of EVs. What can we do to increase our production capacity, and where do you think we need to go in terms of the culture methods that we're using, or even the separation methods, from your perspective?
Of course, everything depends on the type of the cells that you culture. But I think that now there are a lot of efforts in this bioreactors culture, that in generally small space you can, in a small volume, you have some cartridges with the cells, and if they are willing to grow on the surfaces, it's really nice, especially for mesenchymal stem cells, which are very popular in the clinics. So in the clinical applications, and actually they are the main target to isolate EVs from MSCs for the clinical application. And they, as far as I know, they grow quite nicely on these bioreactor cartridges, which are fully automatic systems with the GMP standards, and you can generate liters of the conditioned media in relatively high speed, and not without the need to have these towers of the culture flasks. So I think this is the future of the systems. Of course, not all cells would like to grow on the surfaces. Some cells just require some modifications. For example, our generally IPS cells are very demanding. They are very sensitive to every change in the surrounding, in the incubator even. So it's more tricky to scale up all this procedure. But I think that these bioreactors are quite interesting now, and many, as far as I know, many laboratories work on it to isolate this in a massive scale EVs, especially from MSCs with these bioreactors, and also these new systems of not only culturing cells, but also speeding up the process of the isolation. With this filtering system, high capacity systems, we also bought some system in our laboratory, and we are trying also to to use it to effectively isolate EVs from really liters of a culture media. So these are two points to scale up. First, this bioreactor approaches, and the second, new devices to isolate in a repetitive way, because this is also important, as we see that there is the diversity and the variability between the particular preps. But this new equipment is dedicated for a GMP with validated isolation of EVs for this scale-up approaches.
Well, thanks for that perspective and for the note of optimism. It looks like there's a lot to do still, but we have some strong hopes for being able to take some of these EV-based therapies into the clinic. So this is very good news, and I think a good way to end our hour here. So I would like to thank you, Ella, for all of your work on this and for your perspectives today. And Eva, thank you so much for joining us and for presenting this work. We wish you both the very best as you continue it, and we'll look forward to seeing new and great things from your laboratory. So thank you, and thanks to all of our attendees today for joining and for the interesting questions and for your attention. So thank you, everybody. Wish you a good rest of the week, and hope to see you again very soon. Take care now.
Thank you. And thank you very much once again for having us with you.
You're quite welcome. Thank you.
Thank you, guys.
Thank you.
Bye-bye.
Take care. Bye.
Bye.